The recognition that cells can only come from pre-existing cells was first proposed in the mid-1800s by Rudolf Virchow, a German pathologist.
This was an extension of the assertions by fellow German scientists Matthias Schleiden and Theodor Schwann. They said that all living things are composed of cells. But cells reproduce or divide in various ways. Mitosis produces genetically identical cells for cell growth and replacement.
In animals, meiosis produces gametes, which have half the number of chromosomes as the parent cell. Diversity among organisms is a result of meiosis and the union of these haploid gametes during fertilization. The continuity of life is based on these principles: the strength of similarities
and the possibilities of difference. In this program, we will be comparing mitosis and meiosis. Students will be using chromosome models to illustrate these processes, and you'll also see how the light and electron microscopes are used to visualize the different phases of mitosis and meiosis.
And by the end of the program, you'll have an understanding of where these two processes occur, the behavior and resulting number of chromosomes, the types of cells produced, and the significance of mitosis and meiosis to the continuance of life.
Today we're going to compare the two cellular processes of mitosis and meiosis. But let's start with some definitions. Jason, how would you describe mitosis?
In mitosis, one cell divides to make two cells, each with the same number and kind of chromosomes as the parent cell.
Good. And Shailene, meiosis?
In meiosis, one cell divides twice. The first time in meiosis 1 and the second time in meiosis 2 to produce 4 cells, each with half the number of chromosomes as a parent cell. So we have a difference already.
Right. Mitosis produces 2 cells and meiosis produces 4 cells.
The cells produced by mitosis are diploid. Those produced by meiosis are haploid.
You have to be careful, though, Shailene, because in some organisms, haploid cells divide by mitosis to produce 2 identical haploid daughter cells.
So the way Jason said it is better. One cell divides into 2 cells, each with the same number and kind of chromosomes as the parent cell.
Yes, that's more accurate.
Meiosis, followed by mitosis, results in the formation of egg cells in flowering plants. 4 haploid cells are produced by meiosis in the ovule of flowering plants, but only one will remain functional. This functional cell will undergo mitosis, developing into an embryo sac consisting of 7 cells with a total of 8 haploid nuclei. One of the cells containing a single haploid nucleus will be the egg cell. During fertilization, one of the sperm nuclei that travel down the pollen tube will unite with this egg cell to form the diploid zygote.
Right, here's your assignment. Using these materials, I want you, Jason, to illustrate the process of mitosis. And Shailene, you can illustrate meiosis.
What's the starting chromosome number?
Oh, we can use 4.
Like the same as in 2 homologous pairs.
Right. These processes are a lot easier to understand with a small chromosome number. All right, go ahead.
Well, let's let this wool represent 4 chromosomes in a cell.
Then in interphase, each one duplicates, forming sister chromatids, which are attached by centromeres, represented here by marshmallows.
In prophase, the centrioles migrate and the spindle fibers would form and move towards the center of the cell.
Then the chromosomes also migrate towards the center of the cell until they line up in the middle at metaphase.
And the spindles attach to the centromeres. How are we doing so far?
Oh, good. There are some other events that occur during prophase, such as the fragmentation of the nuclear membrane. But I want you to concentrate on the actions of the chromosomes.
Okay. In mitosis, the sister chromatids line up along the equator in the cell, waiting to be separated. And what's happening with yours?
In meiosis, the homologous pairs line up in a group of 4. They're called tetrads.
But why are you cutting them?
I'm going to be cutting and pasting them to represent crossing over. The chromosomes of the tetrad cross over, resulting in an exchange of genetic material between homologous chromosomes.
Right. Crossing over is a good way to add variation to the cells that are formed by meiosis. We've just witnessed a fundamental difference between these two processes. Can anyone explain it?
In mitosis, paired sister chromatids line up along the equator of the cell.
In meiosis, homologs form tetrads, cross over, and then move along to the equator as well.
You know, crossing over is interesting, but does it occur in mitosis?
Good questions, Jason. You should consult a specialist.
They actually don't cross over in mitosis itself, but a cell that's undergoing mitosis, remember, most of its time it's spent in interphase. And during interphase, chromosomes can cross over with each other, but it's an exceedingly rare event. It's probably 10,000 to 1,000,000 times less frequent. As to the question of whether chromosomes always cross over in meiosis, there are some species that don't have any crossing over at all. For example, male Drosophila don't cross over. But the vast majority of species do have crossovers at meiosis, and when you do, you have to have at least one crossover per tetrad. Because what happens is, in mitosis, the chromosomes line up on the metaphase plate, and each centromere has attachments to the spindle pole. And you have equal force pulling in each direction to cause them to stay at the metaphase plate. In meiosis, since each centromere or each chromosome, the maternal and paternal chromosomes, have their own centromeres, they wouldn't have that equal balancing force. Instead, though, when they cross over, there's something about the crossover point that makes it sticky. So the two homologs are held together at that crossover point until anaphase. If a chromosome does not cross over, then they're prematurely pulled to the pole. They try to find their way back, but the important thing is then they'd be moving independently with each other. And it's possible that both chromosomes by chance will move to the same pole. In that case, you'll have one gamete that has both the maternal and paternal chromosomes and another that has none. So when those gametes are fertilized, you'll end up with either one too many or one too few chromosomes. And for example, if you have too many chromosomes, if it's chromosome number 21, you have what's called Down syndrome.
Not all cells undergo cytokinesis following mitosis. The skeletal muscles of the body are made up of long, multinucleated cells where the cytoplasm is continuous. Mitosis results in nuclear division, but not cytoplasmic division. All right, let's see what else we can discover.
Well, in mitosis, the spindle fibers, which are attached to the centromeres, shorten. As they do this, the sister chromatids separate. Each of the chromosomes then move to an opposite pole of the cell.
Basically, the same thing happens in meiosis, but my chromosomes move in homologous pairs. The end result is that there are the same number of chromosomes at each pole.
Good. What happens next?
Well, cytokinesis takes place. That's the division of the cytoplasm. Are these animal cells or plant cells?
Well, there are centrioles, so they have to be animal cells.
Well, then the cell membrane pinches in, resulting in the formation of two new cells, each with the same number and kind of chromosomes as the mother cell. Mitosis is complete.
What is the phase being represented by the chromosomes moving to opposite poles?
That's anaphase.
Good. And in mitosis, the chromosomes would then unwind, the nuclear membranes reform, and a few other changes take place.
Which is telophase. Both my cells would then go into interphase and perform their cellular functions within the organism.
But my comparison doesn't end here.
Well, carry on, Shailene.
I just need to make a minor adjustment here.
And you'll want some more spindles.
Yes, please. Well, we'll assume that interphase is very short or non-existent. Then there is a second division, meiosis 2. There are no duplications of chromosomes during this division. It is very similar to mitosis, but with one important difference, and that is that 2 new spindles have formed at a right angle to the original one. This happens during prophase 2, when the centrioles migrate and spindle fibers form. During metaphase 2, the chromosomes line up along the equator of the spindle, and the spindle fibers attach to the centromeres. During anaphase 2, the chromosomes separate to opposite poles, this is followed by telophase 2 and cytokinesis.
And 4 cells are formed.
Right. Each with 2 chromosomes.
Which is half the number that we started with. So they are haploid. And you can tell that each one is different genetically because of the crossing over which took place.
And in animals, these cells are called gametes, like a sperm or egg.
Good. If we were to do a comparison between these 2 processes, what would be some of the differences you could list between mitosis and meiosis?
We're already one step ahead of you. We've compiled a list. Mitosis, 2 cells. Meiosis, 4 cells.
The cells produced by mitosis have the same number and kind of chromosomes as the original cell. Those produced by meiosis only have half the number of chromosomes. And all the cells produced by meiosis are haploid.
And tetrads, which are unique to metaphase 1 of meiosis.
In mitosis, there is only 1 division of the cytoplasm, but in meiosis, there are 2.
Good. What about the similarities?
Well, in each case, the chromosomes duplicate, and the relative positioning of the chromosomes are the same at each stage. For example, in metaphase, they're located at the equator of the cell, and during anaphase, the chromosomes move towards the poles.
I have a question. The spermatogonial cells in the seminiferous tubules of the testes undergo mitosis to increase in number, and undergo meiosis to produce sperm. How do the cells know which process to activate?
You know that mitosis and meiosis both happen in the testes. Germ cells undergo these processes in seminiferous tubules. These processes are highly related. It turns out that one cell does not have a choice to go into mitosis or meiosis. It's programmed to go into one or the other type of division. And to explain that a little bit further, I would like to show you this diagram. As you know, in the testes, in the seminiferous tubules, we have stem cells that can undergo a mitotic division and produces a cell which can undergo a number of further mitotic divisions, which we call spermatogonia A1. It's simply designation to differentiate these different germ cells. Spermatogonia can only undergo mitotic divisions, not meiotic divisions. What's also interesting is that the daughter cells that evolve from a parent cell after mitotic division are slightly different from the parent. They're equal to themselves, but different from the parent. So if you look after a number of divisions, we've come to a spermatogonium B, as it's called. This B is unlike its parent or its grandparents, or even further down the road, the spermatogonium A1. B is a special case because it can undergo only one more mitotic division, and that leads to the production of spermatocytes. These spermatocytes are the only cells in the testes that can actually undergo meiotic divisions. They do so, and upon doing this, meiosis means reduction division, as you've discussed, and the result is therefore a spermatid, a round cell, which is haploid in its DNA content. It has one chromosome of each. The spermatid is literally round and must undergo quite a different change in its shape, which takes a long time, to to produce sperm. So mitosis only occurs starting at the stem cell all the way down to this B cell where the last mitotic division occurs. Then spermatocytes undergo meiosis. The real trigger is at the level of the stem cells. All these other cells seem to know what they are doing or what they should be doing, and once this program kicks in, it keeps going until you wind up with a number of sperm. Here we have a seminiferous tubule, schematically drawn, with its membrane, and as you know, the germ cells are present in various positions in the seminiferous tubule. A Sertoli cell, which is a resting cell that does not divide, is present in tubules and is a nursery cell for these different male germ cells. It is a Sertoli cell that receives stimulus from the outside. This could be male sex hormones, or it could be follicular stimulating hormone, also called FSH, and it knows what to do with these signals from the outside and relays that to these germ cells, and the germ cells then respond with a certain action.
Suppose I told you that the process of mitosis could no longer exist. How would that affect living organisms?
Well, the first thing I think of is growth. And growth is the addition of new cells, and these cells are produced by mitosis.
That's true, but I've stopped growing. Wouldn't affect me.
But even though you've stopped growing, you still have to replace lost cells. Red blood cells, skin cells, and the cells that line the intestinal tract have to be replaced daily, and this is done by mitosis.
And there are cuts to heal, broken bones to repair, and the fertilized egg develops into a multicellular organism by the addition of cells. Mitosis is involved here as well.
We couldn't survive without it. Unicellular organisms use it to reproduce quickly under ideal conditions. It's called binary fission.
I don't think any organism could survive if mitosis didn't occur.
Good. What about meiosis?
Meiosis is associated with sexual reproduction. I suppose lower organisms would be all right because they can reproduce both sexually and asexually.
However, higher organisms wouldn't be able to reproduce.
Now, besides forming haploid gametes, which then later unite to form a diploid organism, how else is meiosis important for sexual reproduction?
It has something to do with providing variation in the species.
And I seem to remember something about 2 to the nth power as representing the possible number of chromosome combinations in gametes, but I don't really understand all that.
Well, perhaps I can make it more clear using these materials. Why don't you each take a set of spindles? A meiosis is usually illustrated using 1 pair of chromosomes, but I've got 2 pair here, different lengths. Now we'll let the blue wool represent the male homologue, and the pink wool represent the female homologue.
Okay.
Okay, so what's happening now?
They're duplicating.
Right. Shailene, if you can just pinch them in, we'll imagine that there's a centromere at that location. Good. Now what's next?
A metaphase. That's where the chromosomes line up along the equator between the poles.
Here's a set of chromosomes for you, Jason. Why don't you line them up?
Is there a set pattern?
No, you can use any pattern you like. Okay, now let's separate the chromosomes in anaphase 1. Okay, what combinations did you come up with?
Well, we both came up with different combinations.
Right, that's exactly what happens in nature. We've produced 4 possible gamete combinations without crossing over. And that would be at the end of meiosis 2. We've only shown to the end of meiosis 1. How many pairs of chromosomes did we start with?
2 pair.
And what was your formula, Jason?
2 to the power of n.
Where n represents?
The number of homologous pairs of chromosomes.
Right. In this case, it was 2. So 2 to the power of 2 is 4. Imagine the number of possible combinations there would be when we have 23 pairs of homologous chromosomes.
Now I know why we need a formula for all this.
Okay. You've looked at some of the similarities and differences between mitosis and meiosis. You've talked about where the processes occur, the number of cells produced, the behavior of the chromosomes, and the significance of each process. Now, there's one skill left. You have to be able to recognize chromosomal behavior by looking at an actual slide of mitosis or a micrograph of meiosis. Let's go on a little trip.
How little?
Well, I was thinking microscopic.
Well, normally I wouldn't shrink from a challenge, but in this case, I guess we have to. Let's do it.
Okay, but before we go, we have to be clear as to what we're going to see. We have to pay attention to the behavior of the chromosomes and the final number of chromosomes at the end of the process. What should we start with?
Well, we already have an allium root tip set up and ready to go.
Okay, let's go for it.
Welcome to the cell cycle and mitosis.
Oh, neat stuff. But remember why we're here, and that is...
To look at chromosome behavior. But I don't see any chromosomes in these cells.
That's because we're looking at interphase. Mitosis hasn't begun yet. During interphase, the chromatin material duplicates.
And we can't see the chromosomes until mitosis begins.
Look! The envelope around the nucleus is disappearing.
And the chromosomes are starting to become more visible. They seem to be clumped together.
Right. We're looking at prophase. And we can see the chromosomes for the first time because the chromatin material is beginning to condense. What do you see now, Jason?
The chromosomes are in the center of the cell.
This is metaphase. You can see the spindles really well now.
And here's a cell where the sister chromatids are separating at the centromeres and moving to opposite poles.
Each chromatid is now called a chromosome. This cell is in anaphase.
Now we just need to find a telophase and cytokinesis.
This is a plant cell, so let's see if we can find a cell plate forming between cells. There's one, just above it.
Oh, well done. That structure can be difficult to find. What about the cells on either side of that cell plate?
A new nucleus is forming, and new nuclear membrane is beginning to appear. That's telophase. 2 new cells have been formed, and each has the same number and kind of chromosomes as the original cell.
Okay, let's look at meiosis.
Here's a cross-section of a testis. We should be able to see meiosis on this.
I can't make out any detail. Can you magnify it anymore?
It's up as high as it can go.
We'll need a technology with a higher magnification to look at meiosis.
An electron microscope?
Yes.
What is an electron microscope?
An electron microscope is a research instrument that's used to analyze microscopic information, and it varies from a light microscope in that it uses electrons to do the imaging. This particular microscope can actually magnify to 250,000 times, but what's more important than the magnification is the resolution of the instrument. Basically, resolution, if you think about a light microscope, it can resolve things to approximately 20,000 nanometers, whereas the electron microscope can resolve things to 20 nanometers. So your factor of improvement is approximately 1,000 times.
How important is this tool in the history of biological research?
We're actually imaging something that prior to the electron microscope was never visible as an entity in any other form. We do research on double-stranded DNA, interacting that with protein units called transcription factors. And with this particular instrument, we can actually image individual strands of double-stranded DNA. Prior to this time, no light microscope or any other form of visualization could image to anything close to that particular resolution.
Now that we've learned about the electron microscope, let's use it to study micrographs of the testis. Are you ready? Okay, hang on. Now, what do you notice about this primary spermatocyte?
The nuclear membrane is widening, and the chromatin is condensing.
Anything else?
Well, the endoplasmic reticulum appears to be widening in the cytoplasm.
Good. Okay, now what do you see in this primary spermatocyte?
The nuclear membrane has disappeared.
And the Golgi complex is quite evident. Probably lots of cell activity.
Now, moving onwards, can you suggest what's happening here?
Well, I can see two distinct chromosomal areas, with mitochondria surrounding each one of the chromosomes.
Good. Now, what phase of meiosis do you think we're in?
The end of meiosis 1. And those must be the secondary spermatocytes.
I can see spindle fibers. And the endoplasmic reticulum seems to be changing to form wider structures.
Good. Now, what about that nucleus over there that seems to be smiling at us? Do you know what that is?
No.
That's the nucleus of a sustentacular cell, with its cytoplasm surrounding and supporting all the cells that we see. It functions the same as the Sertoli cells of the mammalian testes.
Wow, this sure is different from the two-cell stage we observed at the end of meiosis 1.
It certainly is. Can you describe what's happening?
I can count 1, 2, 3, 4. I can count 4 cells, which appear to be joined, and the chromosomes are surrounded by mitochondria, as we've observed in previous stages.
And that one, that would be 5. What is that?
Oh, that's the cytoplasm and organelles which must be discarded in order to make the spermatids more compact. Okay, one more and we're out of here. The newly formed spermatids are undergoing final development and differentiation. The mitochondria are surrounding the condensing chromosomes, and the cells are much smaller than the primary spermatocytes.
And the cells are haploid now, compared to the diploid cells that we started with.
Scientific research into the processes of mitosis and meiosis continues. By studying what controls cell division, we may learn how to control cancer cells that divide unchecked, eventually killing the whole organism. Why some cells can repair themselves and others cannot remains a mystery. If we could learn how to control the reproduction of cells, we might be able to stimulate nerve cells to grow. This would enable us to treat damage to the central nervous system.
Our study of mitosis and meiosis is far from over. There are still plenty of unknowns to keep researchers busy well into the future.
Such as solving the mitotic patterns that have gone out of control in cancer cells and bringing about a cure for this disease. Many other diseases and conditions also result from cell division that does not proceed normally.
The ethical impact of these decisions must be given serious consideration.
Many of the secrets of mitosis and meiosis are yet to be discovered, but one thing is certain. Both of these processes are an essential part of all life on Earth.